This video is an instructional video on how to do lab number 5. Some of the people may have looked over it and panicked, oh, I can't buy licorice, or I don't eat licorice, or something like that. So I wanted to talk about that. So to get started for this, you'll notice there's several files. One file is for the instructions. Another file is for typing in all your answers. And then there's the link for actually submitting it. So what you're going to want to do is certainly download the submission file. And if you're actually going to make the model DNA with the licorice and marshmallows, you're going to want to go ahead and get those ingredients. But you don't have to do that to complete the lab. You can work your way through the lab sort of pretending that you have those things. So I just wanted to mention that. Some people have kids that like to help with it and then eat the results afterwards. Other people are hands-on, and this kind of gives them a feeling of how this all works. Now, when you get into start filling out completing the lab, you notice there's some tables in there, and they're in some of those boxes. You're going to write complementary bases. So you'll make one strand of DNA and you'll write that in the box, or you'll pretend to make that piece of DNA and you'll write the various bases, one base per box. And then in the box below them, you're going to put the complementary bases. And if you have listened to the other videos and read your book, you'll know that if you have a T in one box, the box below it's going to have an A in it, because the T and the A are complementary. And so when you have a T in one strand, you'll have an A in the other. Same thing with C. C in one strand, you'll have a G in the other, and vice versa, that sort of thing. So here's an example. You made your DNA. This was the strand you got. Let's say that is the strand you got. I don't know if it is or not. Or you looked at the instructions and said, okay, this is what I'm supposed to make on one strand, so on the other strand, I would have this. See, wherever I had an A, the complementary strand has a T. Where I had a C, complementary strand is a G. Well, after you get through doing that, it asks you to further work with this molecule of DNA you have, and they want you to make the messenger RNA strand that is complementary to one of the DNA strands. Okay, one of the DNA strands, actually they're going to have you do both, but then you're only going to work with one of the messenger's RNAs, and you need to figure out which one, and there's some clues as to which one you need to use. You don't just pick the one, oh, I like this one better. There's got to be a reason for it. So what you're going to do then is you're going to fill in, for the messenger RNA, you're going to put the bases that you have from the DNA in groups of 3, which is a codon. And here's an example. Let's say one of your codons was 3 Cs. Well, then you're going to go in and look that up using the genetic code table to fill out what's in this box. Okay, so whatever's in this box, you go to the genetic codes table and it will tell you what it is. So if I go there and look for CCC, I'm going to find that it's proline that goes in the box. Now, it's easier to go ahead and use a 3-letter abbreviation than try to write that entire word in the box or type the whole word. So 3-letter abbreviations are certainly acceptable. There is a standard set of 3-letter abbreviations. If they're not on your chart, do your best, and hopefully I'll understand what you're saying. So when you've got the DNA and then you need to make RNA from it, you're going to also do that little bit with the complementary bases, but there's a difference. If you have an A in the DNA strand, when you get to the messenger RNA, you'll have a U that's complementary to the A. There's no T in RNA, but there is a U. C and G, they're fine, but there is that difference right there. Then there are questions in between all these boxes that you're filling out. You can be looking at some mutations and the effect of them. So be sure you answer those questions, and the answers can be found either in the textbook or they're in the videos that have been posted. When they tell you to use the genetic code, here's that table I was talking about. And you can see right here, there's that CCC that coded for proline. So if you look at the genetic code table here, you can see there are, in some cases, is 4, actually in this case 6, codons that code for the same amino acid. One of the important ones is AUG. There's only one codon for it, and this happens to be the very first codon of a gene that's going to be, or messenger RNA that's going to be translated. And we also have some stops here that tells the enzyme ribosome you don't need to make any more of the protein. You don't need to attach any more amino acids. So it's real easy to find each of those 3 codons, or each of the 3 bases for the codon in which amino acid it codes for. And this one, this particular table here, already has the amino acids in their 3 letter configurations, which is good. So the other part of the lab, actually a fair portion of it, is looking at the effect of mutations. Now, I created these set of problems to work through, and so they are artificial, and I essentially put a mutation on some of them in every single codon. That's not what you would normally run into, but it's to, instead of having you do a bunch of different ones, having you do just a few strands where everything gets mutated, and you look at you're going to look to see what the results are from that. And then I'm going to ask you to compare. Okay, you had the last base of the codon changed. What are the results from it? Or we changed the middle one, or we changed the first one. What is the effect, and how does that compare depending on where we have that mutation? So you're going to be looking at mutations in the first, second, or third base. I actually have you start with the third and work backwards, just because I like to. Anyway, so when you get to the questions about the effect of the mutation, it's with that particular mutation, did you actually see a change in the amino acid? Or maybe you didn't. That's the kind of thing I'm asking for. And if it did change, do you always get a change, or just some of the time? And you'll see that when you're looking at some of the problems that, oh, yeah, I get a mutation some of the time, and other times I don't. So that's something to keep in mind.